Hey guys, I am Dr. Michael Cavanaugh. I'm in Saddleback Animal Hospital in Tustin. I also work here with the MiDOG group. I've done a lot of sampling for them for 3.5 years. This is Dr. Unita Krumbeck, PhD in microbiology. She kind of directs MiDOG, and she has a fantastic presentation that kind of explains everything about the next-gen sequencing of the MiDOG test.
Okay, well, thank you, Dr. Mike. So it's not just going to be about MiDOG, but really a lecture of where microbial diagnostics are going to go in the 21st century. What are some of the new techniques that you can apply in your clinical everyday life? And what are the new options and possibilities that next generation sequencing has for the diagnostics of companion animals? So, okay, this is not working. That's a great start. Here we go. Okay, I don't want to make this a long introduction about microbial history, but just a quick reminder that the field of microbiology is actually not that old overall. The first idea that something like a microbial cell exists has only been discovered in 1665, and then it took another 250 years until Robert Koch could actually put together that bacteria are causing disease when he wrote his 4 postulates. And with him, really the golden age of microbiology started. And a lot of the techniques that we're still using today very frequently were developed at this time in the late 1880s, such as the agar media, the gram staining, or the petri dish. But the real push for microbiology came with the discovery of the DNA helix in 1953. And that really allowed us to detect more the DNA of the bacteria that were present. And another big push was by Carl Woese, because he discovered the 16S RNA marker. And with that, he found that there are actually 3 kingdoms of life. There's this whole other kingdom of life that we had just no idea that it existed before, because most of these organisms were not culturable. And with that, the first notion came around that there is this microbial paradox, that there are all these organisms whose DNA we can detect, but we cannot grow them in the lab. So they called it viable but not culturable bacteria. That improved a little bit with the development of PCR, still very frequently used in microbial diagnostics today. But what really gave the last push in microbial diagnostics and microbiology in general was the development of DNA sequencing, and specifically the development of next generation DNA sequencing. And the term can be a little misleading because this was actually established in 2005, and it allowed for the massive parallel sequencing of all kinds of samples, and it enabled cool projects like the Human Microbiome Project or Ocean Sampling Day or MetaSub, where they're sampling the subways of all mega cities around the world. And with that, we know that there are 10 to the 20 microbes on Earth now. It's a huge number. And each and every one of us, in our intestinals and our skin, there are 100 trillion microbes for each and one of us. And because we can detect their DNA, but we cannot culture them. So there's an estimate that 99% of all microorganisms around us are not culturable. Yet they have such a big influence on our health and supporting our immune system or being pathogens. And we have this dilemma of the microbial diagnostics. We have all these causative agents that may not be known because culturing can only detect 1% of all of them. Yet we have more than 90 million patients afflicted with infectious disease annually in just the USA. And these chronic infections are often multifactorial, including bacteria and fungi and viruses and parasites. And most often broad spectrum therapy is applied, but that has a lot of consequences. It can lead to potential collateral damage. Because the symptoms for each individual may persist, it can lead to chronic outcomes, and it creates these superbugs. So bacteria that are resistant to every single antibiotics that humankind has developed up to now. And there are several bacteria out there that already are resistant to every single one of them. And by using these broad spectrum antibiotics, it accelerates the antibiotic resistance development across all kinds of bacteria. And it also eliminates helpful bacteria that were present on the area before and were protecting the host from other pathogens by niche competition between the organisms. So there really is a need for empirical treatment. And successful treatment of any kind of microbial infection really starts with the accurate pathogen identification. So you know who's there, who's causing the infection, and then based on that, how do I treat the patient? Yet the most currently used diagnostic tools are still bacterial culture. And you remember from before, those were developed in the 1880s, and they haven't changed that much since then. We have developed some more media, but still, we have 99% of all organisms that cannot grow in culture. So even if we double or triple or even 100% improve our culture media, it will never be enough to capture all of them. It is not only the oldest method for microbial detection, but it's also worldwide used the most. And multiple tests may be required when you have aerobic organisms and anaerobic culturing and fungal culturing. And especially fungal culturing can take up to weeks. So you usually don't have time for that in a clinical setting. And so many culture tests return with no growth. And depending on what kind of sample you have, it can be 10 to 30% of the samples, or for urine even higher. And I will show you some more data about that in a little bit. The other diagnostic tool is PCR, also relatively old, but the great advantage is that it's super fast, it's very reliable, it's super sensitive, and very specific. And what you do need to know, you have to have a priori knowledge of what you're going to target. So you have usually between 8 and 20 targets, and if the results come back negative, that means you only looked at 8 or 20 targets, but there's so much more, right? So once the results come back negative, what does that really mean for you and your patient? And then it also cannot discriminate between causative agents if you have multiple positive hits. And the next generation sequencing, or short NGS, solves a lot of these problems that we see with the other techniques, because it detects all microbes in the samples. If it's a fungus, if it's a bacteria, even viruses, depending on the test, it's very fast and reliable, just like PCR. And it can also detect biofilms, so very complex samples where you have a very high number of different organisms that you don't know about. It can discover novel organisms because there is no prior knowledge required about the organisms that could be there, because it's not target-based. It just amplifies everything that is there. It offers species-level resolution, so it's very specific still. And it can identify viral factors, antibiotic-resistant markers, and microbiome responses to pathogen, because you can basically amplify an entire microbiome. So a very complex ecosystem, you can see how does it respond if a pathogen enters it, or how does it respond if an antibiotic treatment is applied, or any other kind of treatment. And then you can surveillance the changes in the microbiome over time, because it's so fast and also relatively cheap. And it really has been a response to our microbial needs. Over the time, we saw the development first of more media, then PCR, and now next generation sequencing, because I start identifying more and more bacteria, more and more fungi that influence our health. And we needed the technology to catch up to actually treat those in a clinical setting. And it allows to move away from the one test, one pathogen paradigm towards multiplex approaches for infectious disease diagnostics. And therefore it can really help revolutionize clinical diagnostics and ultimately improve patient care, which is, of course, the most important part. It helps with the targeted antibiotic treatment to move away from this one-size-fits-it-all antibiotic treatment, because you know exactly which bacteria are there, which ones will respond to a given antibiotic, and do I even need an antibiotic? Is it maybe a viral infection, or is it a fungus? And with that, it can help to prevent those superbugs that I was mentioning earlier. And I'm not sure if you know, the American Society of Microbiology is in the US the largest collaboration of scientists that get together and review regularly where the field of microbiology is moving, independent even of clinical diagnostics, just anything we could know about the oceans or the Himalayas, anything. So they are really the expert in microbiology. And regularly they produce these reports to announce where the field of microbiology is moving. And in 2015 already, they said that next generation DNA sequencing has the potential to dramatically revolutionize clinical microbiology by replacing all the current single tests within one inclusive test. And it will be the gold standard for diagnosis in infectious diseases. And it's already being applied to humans as well. There are 2 companies currently out there, Microgen and Precision Biome, that are applying next generation sequencing to clinical diagnostics. And I keep mentioning next generation sequencing, and I just want to go over very briefly what it is. Like I said, it's not that new anymore. It was developed in 2005. The first sequencer became publicly available. And what it basically means, you have your genomic DNA. It can be any kind of sample. If it's a pure culture or a fecal sample or a urine sample, it doesn't matter. You have all those different organisms in there, and you break all their cells open. You can do that by sonication or by beating them or chemically. As long as there's DNA, you can break it open. And then you fragment the DNA into smaller pieces. You tag them, each piece, and then you massively amplify. You make multiple copies of them in a really large scale, and then you start sequencing. Everything in parallel. So it's really fast and very reliable. And there are a lot of companies, a few companies out there that are selling this. And one of them you might know is Illumina. They're located close to us in San Diego. So it's a very standardized technique that's used by scientists on all kinds of samples, from the oceans to our pets, to forests, to ourselves, in agriculture, wildlife, and highest points of Earth on the Himalayas. And it's become really the standard method for any kind of microbial analysis that scientists might conduct, with more than 7,000 publications a year. It's hard to keep up with any of that research. And it allows us so much more insight into how we as hosts or our pets as hosts interact with the microbiome and the environment and diet. And that all that is at an interplay and constantly influencing each other. And NGS has really allowed us to understand so much how bacteria influence our health, from anything from metabolism of therapeutics, from the development of the immune system, how we are fighting against pathogens and all other aspects. And specifically for animal health, there are multiple studies, more than 3,600 this year up to now actually, that look at all kinds of animal microbiome. Even in areas where you previously wouldn't necessarily think you would culture, such as the oral microbiome or in periodontitis, and also urine samples and all kinds of samples. And with that, we now have a pretty good understanding what a clinically healthy animal looks like. Here as an example for dogs, what the skin microbiome looks like, and it's actually relatively similar to humans. There's a huge diversity between animals and between body size. So all the way from the nose, the ears, or the paws are all very different amongst each other. But the main phyla are proteobacteria and firmicutes. And these microbes help the dogs by providing a barrier against pathogens, by preventing hypersensitization against allergens, and interacting with the host cells to impact cellular function and immunity. And if you look at this dog, so every pie chart that you see here represents all the bacteria at a given location. And the larger the pie piece, the more of that specific genera is present. And overall, you can see there is quite a large diversity across this dog. But of course, they're not only bacteria, they're also fungi. And you can see here that the diversity is a little less than the bacteria. And the main phyla we find are Ascomycota, which are all colored in this green shade. And because we now have this understanding of all the microbes that are present in just one animal, we know it's actually as diverse as the desert and the jungle. So if you take all kinds of species from the desert, plants, animals, anything included, just from the nose to the foot, it's as diverse as traveling from the desert to the jungle. That's how diverse the microbiome is on our pets and ourselves, actually. Another really great advantage of next generation sequencing is that you can discover biofilms. So biofilms are highly complex microbial consortia, and they are a huge health burden, both for animals and for humans. And according to NIH research, they're causing 80% of all infections. And by most culture methods, you cannot detect them. Because what a biofilm is, it starts with planktonic cells that are floating around, finding a decent surface where they can attach and start forming these first microcolonies that then keep growing to mature biofilms. And the top of the biofilms then disperses and starts the cycle over. And of this whole cycle, only the planktonic cells, only the floating cells are the ones that are able to grow in culture. But if we zoom in specifically here what a biofilm looks like, you can really see it's a living 3D structure. It's like a mega city in itself because it consists of all these multi-species consortia. It's encapsulated in this matrix of polysaccharides and proteins and DNA. And you have this gradient of oxygen going inside and gradient of nutrients. So on the outside, you have cells that are growing pretty fast. And on the inside, because there's not much oxygen, not much sugar left, you have these slower growing organisms. And they all interact with each other. They have signaling molecules. And this is one of the main reasons why it's so difficult to treat them. You have the extracellular matrix that's negatively charged. So positively charged antibiotics only can get to the surface of it, if that at all. And the ones in the center, they never even see the antibiotic. They think it's like nothing ever happened. And even if you can attack those cells on the outside, the ones in the center still might be completely unaffected. And once the outside cells are dead and the treatment is stopped, the center cells start growing up and you still have the recurring infection. And because there's such a huge diversity of different organisms there, it is very likely that at least some of them are already resistant to whatever kind of treatment you might apply naturally or by different genes, by mutating them along them. And this, of course, is a very it's a highly pathogenic system. And yet it can never fulfill Koch's postulates because most of them will not be able to grow by themselves in a lab. And therefore, it's very difficult to find all the members that could be in a biofilm and to effectively attack all of them to prevent a reoccurring infection. And when we look at this resistance to antibiotic compounds, there are two types of antibiotic resistance that we can look at. One is intrinsic resistance, so innate ability of those bacteria to not be killed by an antibiotic. So they either have no target for the antibiotic, or they might be able to just take up the antibiotic and degrade it, or they can use efflux pumps to get the antibiotic out. And so it's just not effective because the antibiotic basically has no target in this bacteria. The other one is acquired resistance. So this is based on genetic mutations or horizontal gene transfer or plasmids that are moving along. And these can be easily shared between different bacteria. This is work done by a great group in Germany, in Heidelberg, and they look how antibiotic resistance is easily spread among microbial communities. So if you imagine you have a bacterial cell that is already resistant, it has these little jumping DNA plasmids that it can easily transfer to cells, like the blue ones that were not previously resistant, either by making physical contact and transferring the DNA over, or those cells can take up free plasmids that come out if a cell lyses, or also by bacteriophages. And this happens really quick. Remember, the generation time of E. coli, for example, is only 20 minutes. So once one E. coli cell acquires this, it can spread easily amongst them. And this actually works across gram-positive and gram-negative organisms as well. And that you can really trace these resistances on a genome-wide scale was shown by this group, by Dr. Jordan and all. And they used a clinically relevant pathogen, Staphylococcus aureus, a very important human pathogen. And they had 500 isolates of this organism for sequencing and 491 for culturing. So they couldn't culture quite all of them, but most of them. And for culturing, they tested phenotypically like you would do for any of your samples. They send it to a lab and they test it for all these antibiotics and see does it grow or not. But they also sequenced all these organisms to a very specific scale, and they could trace, is this organism going to be resistant or not? And they found that the sequencing approach was as sensitive and as specific as routine antimicrobial resistance testing, and that generation sequencing actually holds significant promises for the resistance prediction in Staph aureus and other samples as well. But of course, for you in your clinical settings, you're not going to sequence all of that, and you usually don't have isolates. What normally happens, you have your culture sample, you have an isolated bacterium, you test directly for the antibiotic resistance phenotypically, and then NGS on the other side uses any kind of complex sample because it doesn't need to grow, such as biofilm, feces, or urine, and it tests for genes instead of the phenotype. So it uses the genotype to predict the phenotype. So let's see what that looks like in an everyday setting. Say you have a patient with an infection, and it has a consortia of different organisms that could be potentially causing the infection. You send a sample for culturing, a few colonies grow, and you select a single colony, maybe one or two, to do antibiotic resistance testing. And it's based on the assumption that this single organism is the same as all of these ones, meaning that the bacterial species that come from the original sample are all monoclones. They are all basically the same. They're all identical. And then the result comes back, okay, this organism is resistant to 1, 2, 3, 4, 5, and it's sensitive to the other ones. And then you look, okay, now I know which antibiotic to use because I know from this one, so I'll assume it will be the same for my patient. And for intrinsic resistances, that does apply. So for the same species, it will be the same. They don't have the same target. They might have the efflux pump or the other mechanisms that I just explained. So for that, it is applicable. But once it's an acquired resistance for mutation, gene transfer, or all the other things, that doesn't necessarily apply for all the bacteria that were present in your original sample, because you might have picked this one, or this one, or this one. So it's not a homogeneous or monoclonal sample in that sense. So the difference with NGS is you have, again, your patient with all these different resistances and different bacteria, and you completely skip the culture testing. You go straight to sequencing, and that will spit out who is there and how many of each are there relative to each other. So we see most of them are yellow, okay, more yellow than the other ones. So it gives you a pretty good estimate. And it will also depend, so we know who is there, so we can go back to databases and predict the intrinsic resistance, because this is all published data that you can just pull from other publications, and reliable. It's being updated constantly because so many groups are working on this worldwide. And then you also can detect the resistant genes. You can see different genes that you identify and how many copies of those do you have. And because you know who is there, how many of them are there, and how many resistance copies I can find, you can trace them back and align them. And then you can say, okay, in my pool, some of them have these resistances, others not, but because some of these yellow ones have the green resistance, I better not use that antibiotic because these ones will die, but not the one that has the resistance. So let's look at a case study, specifically what happens in an individual. So here we had a dog that had a chronic ear infection. It had been previously treated with beta-lactamase antibiotics, and the infection was persisting. It was just not clearing out. So what was going on? We sent it both for sequencing and for culture testing. And sequencing found there's a high abundance of Staphylococcus pseudointermedius, common pathogen in ear infections, so nothing too surprising there, but in a very high abundance. But the resistant gene was found in a very low abundance, very little of it. So it indicates a heterogeneity of this specific species in the ear sample. And culture testing found the same things, pseudointermedius, and it was sensitive. so indicating that it does not have this gene. Yet the infection wouldn't clear with that specific antibiotic. And and NGS could explain it because a subset of the bacteria had it and not all of them. And by chance, it was not picked up by culturing. Another example here is for skin infection. Again, Staphylococcus pseudo intermedius. In this case, a resistant to fluoroquinolones. And NGS said, well, only 19% of the pseudo intermedius have this mutation. So again, a heterogeneity. And culture found the same thing, again, pseudo intermedius, and it was susceptible to fluoroquinolones. And these were real-life cases that we had in our laboratory. And based on that, the treatment was changed and the infection cleared in both cases. But it just shows that if you have your agar plate, and even if you select multiple strains or colony-forming units of the same strain, it doesn't necessarily represent the entire species that is present in your patient, because it is such a huge diversity and there could be so many of them. So you're playing really a game of chance if you're picking this one or this one or this one, what the culture test will return. So using molecular antimicrobial diagnostics really has the potential to diagnose faster than culture testing, which is of course a huge advantage clinically, so you have your results faster. It's more sensitive than phenotypic testing because of the examples that I was just showing you. It can confirm resistance mechanisms that are relevant to phenotypic resistance. It can help with surveillance data for outbreaks or hospital surveillance and so on. A shortcoming is that it cannot detect novel resistances. It heavily relies on these databases, but these are constantly being updated by researchers all around the world, but you have to keep it up to date. And it allows you to measure antibiotic resistance on a population-wide scale instead of just using 1 or 2 colony-forming units. And of course, antibiotic resistance is a huge worldwide concern, and next-generation sequencing can help address it. We currently have about 70,000 people dying of antibiotic resistances in superbugs right now. By 2050, that number will rise to 10 million. That's only 30 years from now. And these superbugs are super scary, and they're very hard to track by culture testing, basically impossible. And all the surveillance data is done by next-generation sequencing. So with that, you already know now that NGS can help with the detection of biofilms, bacteria, fungi, and antibiotic resistances to prevent those superbugs. But how does any of this apply to you and in your everyday life? I really went on a global scale there, so let's bring it back to you in your practice. And I would like to show you 2 examples. A urinary tract infection and a dermatitis case. And with that, I would like to start that dog urine or any kind of urine is never sterile because we can detect all those microbes in there. We find that even urine from healthy animals collected via cysto is full of bacteria and fungi. Most commonly found are Pseudomonas, Acetobacter, and Sphingophobium. So it's never sterile, but yet there are so many papers out there looking at different ways to culture urine. And this is recently published, actually July 2018. And they were comparing if I freeze the urine or if I add some media to it, how likely is it that bacteria will survive and will the antibiotic resistances change and all those kind of things. And so, yeah, refrigeration affected antibiotic colony forming units and sensitivity. And I almost put the paper away, thought, nah, this is not going to be useful for me. But when you look more specifically into their methods, it was like, oh, data were not normally distributed, but a high portion of samples yielded no growth under all 3 conditions. They had 104 urine samples, and these were patients that were in a hospital, in a veterinary hospital with a suspected UTI. All the doctors, okay, this is a UTI, we should send it for culturing. It was not random dogs. And yet only 64% of them showed no growth. So for you in your practice, that means more than half the time you have a patient there that you think has a UTI, and just by chance you send it for culturing 50-50 if you get anything or not. That is pretty drastic. And that was just somewhere hidden in the methods. And urine is never sterile, whether there's an infection or not. And actually, when we look from our data in healthy dog urine, also collected via cysto, we find 10 to the 5 cells of bacteria. So that's 100,000 cells in 2 mils and 10,000 fungi in 2 mils. In healthy urine, all across the board, we looked at hundreds of dogs where our veterinarians would tell us, to the best of my knowledge, this is a healthy dog. But still, it's full of bacteria and fungi, which are part of the normal healthy dog microbiome. But when we look more specifically which organisms we find, the top one is E. coli, then Staphylococcus, Streptococcus, Proteus mirabilis, which is pretty difficult to culture. And we find most of them frequently in healthy and in UTI cases. But there are others like Klebsiella and Mycoplasma, which here are facultatively anaerobe, so relatively difficult to culture. We never find those in clinically healthy samples. But in dogs affected with UTIs, they're almost half of the entire microbiome, or in the case of Klebsiella, almost 100%. So these microbes are there, and they're always there, and just because they are not culturable doesn't mean they could be causing infections. The other example is on atopic dermatitis. They were looking at 14 dogs that had atopic dermatitis and 17 healthy dogs, and they were looking at them right when they're at their sickest, right after antibiotic treatment, like the last day of antibiotic treatment, and then 4 to 6 weeks later. They sampled several areas in the dogs, and they were then monitoring how much are the dogs improving. And they were on antibiotic treatment, so overall the dogs improved. But they were also specifically looking at their microbiome. So this is what the healthy controls look like. And every single bar here is an individual dog, and every different color is a different bacterial genus, and the scale is over here. So we see a little bit of Staphylococcus in the pink, but overall there's a huge diversity. This is what the dogs with atopic dermatitis look like. And even if I didn't tell you who is who, you can already tell there's quite a large difference. There's this huge overgrowth in most of the dogs of Staphylococcus. And that normalized over time. So this is right after antibiotic treatment, and this is at the washout point. And you can see the amount of Staphylococcus did go down, and Corynebacterium here increased a little bit, and overall the diversity in those different samples increased. So they could really measure the relationship with the microbiome and the dogs improving. They can show what impact the antimicrobial therapy had, which microbes responded, which ones didn't. And they can measure it over time to see when is the dog back to normal. Of course, there's not only bacteria, there's also fungi. Here's again what a clinically healthy dog looks like. This is a case of allergic dermatitis. And they found that Saccharomyces cerevisiae is more associated with a healthy dog, whereas these one on the other side are associated more with diseased dogs. So you can clearly distinguish which fungi are causing diseases as well. But overall, there's a lot going on. And here I'm showing the microbiome as bar charts. Earlier I showed them as pies. I personally prefer to show them as donuts. So it can get quite confusing when you're starting to look into the literature. And you have all colors of the rainbow, and what does any of this mean, right? So you can just picture that either this donut or all that is 100% of all the bacteria or all the fungi, depending on what you look at, that's 100%. And the larger the piece, the more relative abundance is of that certain one organism. So it's, in this case, all based on relative abundance. But even Robert Koch would be thinking like, that's a lot of data, that's a lot of bacteria. How do I know which one the pathogen is? What do I do with all of this? Because it's almost feel like fly fishing versus overfishing. With culture and PCR, you get 1, 2, or 3 results. But with this, we usually find hundreds, if not thousands. Even on a clinically healthy dog skin, we find about 250 different species. That's a lot. And that doesn't mean this dog is infected at all. That is just the normal healthy flora. And that's where it comes in to see what does a clinically healthy dog look like? How do we put this into perspective? So in a clinically healthy reference dog, we see a huge diversity. But this is a case of a skin infection, and you can already tell right away without knowing which bacteria those two look different. And those two are definitely overgrowing. And you can see, okay, these two are probably the culprits. Versus when we look at the fungal profile of the same dog, that looks pretty similar. And so you have quite a bit of confidence that, okay, this is a bacterial infection I'm dealing with, and these two are the ones that I need to be focusing on. And when you don't only look at one dog, but hundreds of dogs, and this is actually from a publication we're currently working on, we see generally that a clinically healthy dog is characterized by a huge diversity of both fungi and bacteria. Fungi a little less than bacteria for skin and ear samples. And for a lot of the cases, we see this complete overgrowth or the lack of diversity in symptomatic cases. And while we do know what a clinically healthy dog looks like, and we can give you all that background, okay, this is how much we normally see in a dog, this is what we would expect, this also applies to other species. This was a very recent patient of ours, actually 2 weeks ago. It was Dusty from Milford, Ohio. She's Dr. Dahlhausen's patient, and she had a chronic oral cavity infection. And he said, I see all this cocci under the microscope. I send it for culturing, nothing comes back, because birds have a relatively high body temperature, and culturing doesn't necessarily adjust for these different environments that the sample actually comes from. And we sequenced Dusty, our first bird actually, and we found that there were only 2 bacteria we could identify. These 2, and you could see right away, okay, that can't be healthy. I don't know what a clinically healthy bird looks like, but that does not look good. And the fungal profile was still quite diverse. And of course, we have the antibiotic resistance profile, and Dr. Dahlhausen treated based on that. And so I asked him, well, do you mind that I don't know what a clinically healthy bird looks like? He's like, no, this was very, like, it's just, like, I look at that, I know what to do. It's great to know what maybe would be normal, but in this case, he was able to treat without any question. And of course, these are just our experiences, these are just case studies, but there are hundreds of publications looking at how next generation sequencing can aid infectious disease treatment. There are more than 10,000 publications on PubMed. This year, more than 10,000 alone. You basically can't keep up with them anymore. And I highlighted here a few of those that I thought were relevant to you. And the consensus is that NGS has significant advantages over culture testing, mostly because it really detects everything that's there. And then on top of that, we're kind of using a unique approach that most papers don't currently use, is combining the relative abundances, so the pieces of the donut, with absolute abundances. So we already know who is there, and we know it's a lack of diversity, but it also matters how many are there. And we looked at hundreds of dogs, in this case for ear infections, and we saw that the total bacteria amount is 3 logs higher than in healthy dogs. And for fungi, it's still 1.5 logs higher. So just by the amount of different organisms you find, you can already have a pretty good idea, am I dealing with a dog that's healthy, or am I dealing with a dog that's subclinical or already infected? And we did that for hundreds of dogs. And down here, we list all the dogs that we were looking at, and up here, the top organisms that we find. And every time you find an organism, you give a color. So the more often you find these different organisms in a sample, the darker it will become, and the more abundant they are, the darker the spots. And you can already see right away, there are these clusters over here. And up here in red, those are dogs that have an infection, and the blue ones are the healthy dogs. And you can pretty clearly distinguish that these 2 clusters and some clustering down here is only or mostly seen in clinically diseased dogs. And those are common culprits like Malassezia and Staphylococcus pseudointermedius. Here we have Staphylococcus schleiferi. And this is a collection of different anaerobes. I know you probably, you can't read the scale, so you have to believe me here. And if we look more specifically, who are the most abundant organisms we find, we actually see like Staphylococcus and Malassezia. They're in our symptomatic cases, but they're also in our clinically healthy dogs, but at significantly lower numbers. So here it's at 10 to the 8, and this is only 10 to the 5, so 3 log differences. So just the fact that we are detecting Staphylococcus pseudointermedius doesn't mean this dog is infected. It's just how much is there? Is there bacterial overgrowth? Is it dominating the sample? So just merely detecting organisms does not predict that they are actually the pathogens or are actually pathogenic in these cases. And a lot of them are actually anaerobic. This is just the top 10, and we have Streptococcus canis, a facultatively anaerobe, and Peptostreptococcus carnic, a completely anaerobic organism that's often missed by culture testing. And when we start looking more specifically, well, if we find organism A, do we find organism B or not? And we can start seeing that a lot of them show up together or just alone, like Staphylococcus pseudointermedius. If that's the case, if it's overgrowing, there's nothing else there, or even lack of other ones. But like Phenegolia magna, all these blue ones are organisms that are also high when Phenegolia magna is there. So that hints at the idea that these organisms are always together, and Phenegolia magna is anaerobe. And for example, this one, Streptococcus canis, is facultatively anaerobe. That leads to the idea that sounds like a biofilm, right? Because they show up in all these hundreds of samples always together, and they have these very specific growth requirements. And so you can start tracing that down. And when you send in your sample and we find these kind of things, we can have such a good idea, okay, it might be a biofilm, it might be, this might be causing the infection. So I'm a microbial ecologist. I get really excited about this kind of stuff. But again, what does this mean to you? How does all this apply directly to you and your patients? So we had, again, Heidi, a 13-year-old dachshund lady. She was actually Dr. Kavanagh's patient. And she was facing bilateral ear ablation. She had been treated for Staphylococcus pseudointermedius multiple times. Culture tests always returned that that was indeed a pathogen found. Antibiotic treatment was based on that result, but it just wouldn't clear. And her primary vet sent her over to Dr. Mike and said, I want to hear your opinion before we actually do this ablation. And Mike sent her over to us. And indeed, we also found Staphylococcus pseudointermedius in a pretty high abundance. But we also saw that her fungal profile was way off. Huge overgrowth of Malassezia. And culturing didn't pick it up. I don't know why. Malassezia doesn't take that long to grow. Maybe fungal culturing wasn't ordered. But anyways, Dr. Mike changed her treatment and used an antifungal, and her ears were saved. So that was very exciting. This is another case from Paloma. She's Dr. Michael Morgan's patient in Quail Animal Hospital in Tustin. Also a chronic case that he had been treating for a couple months. He sent out for culture testing. It either came back negative, or if something came back, it came back Enterococcus faecalis, just one isolate. Relatively high numbers, 10,000 to 50,000 CFU per mil, but the infection still, it would not quite clear. So we used our test on this case as well, and we found a bunch of different bacteria and fungi. Every different color is a different species, remember? So this urine was not anywhere close to sterile or just overgrown by one organism. And if we look more specifically at the bacterial profile, we found ureaplasma, also very difficult to culture, and Mycoplasma canis. So Dr. Morgan based his treatment on this Mycoplasma over here, and the infection cleared. But of course, we were curious to, well, if Enterococcus faecalis grew in culture testing, do we actually find it too? And we did, but it was at 0.05 relative abundance. So it's one of these tiny little slivers over here. And actually the species ID was Faecium, not faecalis, but close enough, right? But it was there, but it was just not the major culprit of this infection. And with that, I would like to summarize that next generation sequencing does have some limitations. You really need to determine the clinical relevance of the data. Merely detecting a pathogen does not say it is actually the culprit if it's at low abundances or different from what we normally expect in a clinically healthy dog, or it might be a subclinical sample. You can't detect novel forms of antibiotic resistances. It's relying on database and research being done by other researchers all around the world. It does require a revision of Koch's postulates. It does not apply to the one pathogen, one disease paradigm at all. And there is a learning curve because you find so much more out. You might find novel pathogens that have just not been known before. And so the treatments might need to be adjusted. The benefits are that it's very fast, highly specific and sensitive. All organisms are identified, especially those in biofilms or in complex samples. It allows real-time patient management. So if you give an antibiotic, you can trace how the microbiome is responding to it. And it therefore also avoids these educated guesses or broad spectrum antibiotics because you know exactly who is there and what are they resistant to. It helps with the detection of novel polymicrobial infections such as biofilms. It's completely growth independent, so it can be done right away. There's no need to wait for any growth. And at least for us, there's no cold chain required. It can help with the antimicrobial therapy. So narrow spectrum antibiotics can be used instead of broad spectrum antibiotics or no antibiotics at all if it's a fungal infection, for example. And therefore it helps reduce the pressure of antibiotic resistances, at least on a small scale. It helps avoid collateral damage by killing off the natural flora with inappropriate antibiotics. It can trace the phenomenon of this gene transfers across different settings or across different species. And it is really the new gold standard for human clinical diagnostics. I think because it is so much more information than what clinicians are usually used to, there is such a high learning curve for some that I think that's why it's taking a little bit to take hold. It has significant advantages over other diagnostic tools. It offers the identification of all pathogens for better treatment decisions, and it can aid antibiotic stewardship. And MiDOG is making this technology available for veterinarians. And we uniquely offer absolute and relative abundances of all those microbes. It's very easy. You just swab your patient. You send the sample to us, no cold chain required. We give you a DNA preservative. So no microbes will grow, nothing will decay. It will be just like you saw your patient. You send it to us via FedEx prepaid already. And then you receive our report via email. We have swab collection kits and urine collection kits. And like I mentioned a little bit, we are more than just MiDOG. We know what a clinically healthy dog looks like, but we also run any kind of other samples. We have dolphin samples recently, birds, bunnies, as you know, for example. So we can really run any kind of species, any kind of sample. These are some of our current customers and some dogs that we have helped in the community. And with that, I'll also be happy to answer your questions soon, but Dr. Mike is also going to go over some of his cases and how this technology has helped him.
I'm going to walk you through these cases just so you guys can, I'm going to tell you how I interpret these reports. Because there's so much data. When I first started doing this with them, they gave me all this data and said, here you go. And I said, whoa, what am I going to do with all this, right? And so we kind of refined the report down. And Abby is an 8-year-old female spayed dog that came from the shelter. And the owners adopted her 3 weeks before bringing her into me. She came in with just pus coming out of her ears and a big mass in one ear, and it was mainly infection in the mass. And so I both cultured it and I sequenced it. Culture came back with Pseudomonas, just poured out Pseudomonas. And it didn't have any staph in it at all. It was just all Pseudomonas. And when I got our sequencing back, you can see that it has the Corynebacterium, the Fusobacterium, the Pseudomonas, which was only about 4%. And the staph pseudointermedius which was just under 2%. And it was almost all bacteria. There was very little fungus. And when they were treating the Pseudomonas with cephalexin because it was sensitive to cephalexin at the shelter, but the staph was resistant. And as they treated it, they got more and more staph pseudomonas growing in there. And so as we treated it, it took us about 2 months to treat it. We used like an EKT, aerofloxacin, ketoconazole, triamcinolone, the ear packs. We used that as well as orals. And we ended up using a marbofloxacin on the dog for about 3 weeks, and it cleared it up. The mass that was in the ear got down to the size of about half a centimeter, and I was able just to cauterize it out. And now the dogs, we are 3 months post, and the dog is completely clear, asymptomatic. So, on the report you have in front of you for Daisy, I'll go into more detail now, so you guys have that in front of you. So Daisy is a dog that I've been seeing for probably 10 years. And over the last about 4 years, she's had these chronic UTIs. She had a UTI, I look at it, she may or may not be able to culture it because of finances. If I did culture it, it came back as a staph pseudomonas. I treated it with Simplicep, pretty straightforward. I treated it for 2 to 3 weeks. The dog would be fine for 2-3 months. She gave a secondary UTI. I cultured it one time, came back with no growth, but it was antibiotic responsive. And I went on and did this for like 4 years. And so finally, I sequenced it. Once we got the technology here, I was able to sequence it. And this is what I got back. And when I look at this first page, you see that there's also Enterococcus on there too. I never got that on the 2 cultures I was able to do. I never got that on there. It never came up. There was also some funguses in there. But if you look at the ring at the bottom, you see how it says bacteria 100%, fungus 0%. That told me that this is a bacterial infection. There's no fungal matter there at all. There's very little. That's just either I took it as it was a contaminant going through the skin, when I'm doing my cystos, or is it just cellular debris at the bottom of the bladder? One of the two. Okay. The second one was the antibiotic sensitivities, or genetic resistance. And as you can see, my Simplicep wasn't working on that Enterococcus. So I changed up my treatment and treated that dog hard for 3 weeks with marbofloxacin. And that dog, when was this done? I can't remember exactly when this was done. It was done, I think, in March. I haven't seen the dog back at all. I called her today before we came in. The dog's still asymptomatic. So those are the 2 main pages I look at. But as we get more and more, the following pages, if you go to this one here, bacterial analysis, that kind of quantitates how much bacteria was there. It was only 20% Enterococcus off this one, and 80% staph still. But when I treated for the Enterococcus, obviously I treated the staph at the same time, and it worked really well. And then as far as the next page is the fungal composition. This works a lot better with a lot of your skin cases, and that's like the ear case we got, the Heidi she talked to you about earlier. It was being treated with the antibiotics, and then we found out there was also some lesion in there. When we did a ear swab on that case, you saw fumalesia, which I typically see a fumalesia in the ear. But there was quite a bit in there, and when we treated it, she did a lot better. And then there's just, on the backs of these pages, there's just references. So that's how I've gone through and looked at these reports and how I've treated off these reports. I've had quite a bit of success. I do more and more of this than I do culturing. I'm still culturing because, I don't know, I guess I'm still comparing the two in a lot of ways. Don't tell them I said that, but I'm still comparing the two in a lot of ways. But I'm really leaning more towards the sequencing now than the culturing because I'm getting more information and I'm able to treat a little bit better. I've had a lot of success treating off the genetic resistance rather than just the sensitivity resistance.
So the way we design and break down all the data we find is really on the first page. You just have your standards, your name, the patient name, and all those kind of things. And then the clinically relevant pathogens that we found. And those are based on literature. What are pathogens that we find in this kind of sample? And all these are blue. So you get this as a PDF. All these are clickable links. So if you have more questions about any of these species, you can just click on it and it takes you to a third-party search. So you can learn more about each organism. The little numbers behind are just the citations how we determine it's a pathogen. So then here you have the absolute abundance, relative abundance, and what you would find in clinically healthy dogs. So pathogens first, bacteria and fungi. In this case, no fungal pathogens. And this one really shows, like Mike mentioned, the entire microbiome. How many are fungi? How many are bacteria? So you can see right away, am I dealing with a fungal infection or bacterial infection? And then the second page has the antibiotic resistance for these bacterial pathogens we identified. And we do let you know whether or not it's an intrinsic resistance or if we found the resistant genes. So it's labeled differently, but the color coding is there for everything. And here's the little notes explain it as well. And for most cases, you can probably stop there. That's all you need to know in most cases. If you do want to learn more, or it's a specifically chronic case, or you just have more questions, this one shows you anything that we find interesting in your sample. Because we have hundreds of dogs, we can compare your sample and see anything that's a little unusual. For example, the lack of diversity, a huge overgrowth, we tell you that here. If there are any other bacterial microbes that are there in really high abundance, more than what we normally would see, but they're not quite pathogens or not labeled as pathogens yet, we let you know here. And if there's nothing that raises a red flag for us, this page might even be empty. But then on the next page, this is not my friend today. We go into the detailed comparison between your sample and our clinically healthy database. So we visualize the data for you so you can see exactly what's going on. Again, here all the organisms are listed, all with clickable links. What we find in your sample in absolute and relative abundance and clinically healthy for bacteria. So we give you the top 10, everything above 1%. And then the same for fungi. But for most cases, you might not need this. It's really, if you want to learn more, if you're particularly interested in that case, and then, of course, methods. Do you have any questions for us? I know it's a lot. You can always email or call us too, or go to our website. We're always happy to help, also with the interpretation of the results. If there are any questions, we have Dr. Mike, who's helping with the data interpretation. We have also Dr. Aline Rodriguez-Hoffman from Texas A&M on our board. So if there are more questions specifically on treatment, our vets can help with that too.
How did you select the healthy dogs to compare to the unhealthy?
So that came from vets that we have been working with, mostly from the Southern California area. There were about 80 different clinics. Also, Dr. Rodriguez-Hoffman in Texas A&M, she has a huge collection of dogs too. So these were dogs seen by veterinarians and determined clinically healthy to the best of their knowledge.
The criteria is if they don't have super infections.
No symptomatic.
At 10 years of age or something like that.
Yeah, most of them were adults. We had very few puppies in there. But to the best of our vets' knowledge, these were clinically healthy dogs.
When we chose ours, we did a lot out of our clinic. We chose a variety of ages. We tried to mix it up. And these were all dogs that were either in boarding or grooming, had no lesions, had a nice healthy hair coat, no redness, no erythema, nothing like that.
And they're on a variety of different diets. We have some, well, most of them from California. We have some from Princeton University, really all across the US.
But even those dogs, you would almost have to base a healthy dog with one that has never had antibiotics. Or at least recently.
Right.
Or they'll change your biome. So that's another question I have is how does, say, I am treating somebody with antimicrobials or antifungals, and then I want to do a MiDOG sample, how does that maybe affect the interpretation?
Well, it will affect the microbiome drastically, of course. We will.
If I am treating with antibiotics and then I do this and I get a result, I can then transfer maybe to whatever other antibiotic your testing indicates would be the best.
Yes, exactly. So you will still see what pathogens are there. Have they responded already? It also depends on how long has the dog been on, or patient been on antibiotics, how long has the washout been. So we have a few cases from Dr. Glassberg, actually, his dog San Santiago, I think. Yeah, he has chronic diarrhea and he's been treating with antibiotics and it's like going in waves. And so we're following this dog for several months to see the effect. But as you saw in the skin, for example, it takes up to weeks. You can still see results. And so.
And just out of curiosity, are you finding the microbiomes are different regionally across the world or even within California? You're saying you're testing stuff in California.
Mm-hmm. Well,
in Europe, say?
Yeah, well, we try to cover at least the United States very broadly. We have some from Texas, from Princeton, from the Midwest a lot. And we see mostly it's depending of if there are other animals in the household or if their dogs are only inside or outside too. For feces, diet has a huge impact. And of course, the nose is very different than the skin because it's wet and all those kind of things. So there is a wide variety across dogs and across different body parts. But that's where we started sequencing thousands and hundreds of dogs in collaborations with universities just to get the numbers up. And that's where we have, with a certain confidence interval of 95%, this is what we would consider normal, right? I mean, it's always a range.
For infection, one of the things she mentioned earlier is actually a loss of diversity.
Right.
So basically, you can see different dogs might have different skin microbiome, but it was very diverse. The abundance was not necessarily very high, but very diverse.
Yeah.
So as soon as there's an infection, that's the Staphylococcus infection, also the Morris infection, you see it overgrow and then completely take over the whole community. That's a loss of diversity. That's a common thing, and that's very easy to find. Actually, very easy to find. Actually. Yeah.
Again, out of curiosity, have we determined how that happens? That you all of a sudden have certain bacterial populations overtake, like the interaction with the immune system, or
Like why you get lots of diversity.
Yeah, what is generating? Is it genetic basis, environmental, probably both, as everything seems to be in
I saw I heard of some of them is actually initially might be triggered by allergy and other things, or maybe just scratching, biting the barrier.
I don't know of any papers or anything saying why that
Yeah,
happens.
that's what I'm curious.
Yeah.
Like, get to the root of stopping it.
And I've read a lot of papers on this,
Yeah.
believe it or not. I have not seen anybody find an idea why that happens, but we definitely see it.
Yeah. I think it's the world we're living in. It's much cleaner than it used to be.
Honestly, I think so too.
Even the puppies, just like human children now, are less exposed. We use antimicrobials for everything.
I know, they never touch the ground, right?
Yeah.
I want to go back to your usage of antibiotics before you sequence. You asked about that. Looking at it, when I do mine, one of the things I think about when I do it is I use the antibiotics. Now, when we sequence it, you're not only going to get all the living pathogens in there, but all the ones you've already killed.
Right.
And so you have to take that into consideration.
But that's the relative abundance.
And
Which I have a question about.
actually, once the bacteria are dead, the cell walls degrade and the DNA degrades very fast.
It does.
So even if they are, so the dead life issue is definitely a question we get a lot. But once a bacterium is dead, the cell wall is harvested by all the other microbes. Everything is used up. So dead microbes, we might find trace amounts of them, but they're never there in high amounts. It degrades really fast.
So in regards to this relative abundance and absolute abundance, on the sequencing, it's been years since I did this. I left molecular biology in 1993, so way before all this. I used to be the PCR machine, right? I'd stand there 3 minutes, 5 minutes, 4 minutes, 3 minutes, and then we'd go on all day. Right? So that was one of my day, and then the sequencing shelf up to the ceiling. But in any case, are you timing it? Is that how? It's like how much replication or amplification you get at a certain time point is how you get the abundance?
Well, that's a Xu Chang question right there.
It is by PCR.
Yeah.
Absolute communication. As you now you're not doing it by hand. Now
Right.
there's a machine doing some cycling.
Yeah. Immunex, I used to work for Immunex. They had bought one of the first PCR
Yeah.
sequencers right when I left.
Yes.
I was like, okay, I don't have to do that anymore. But oh well.
Yeah. So basically, you can quantify the microbes by how many cycles you need to reach certain signal.
Okay, got it. You're cycling. Correct. And then the primers must be extraordinary now to be able to get all this.
Correct.
I used to have to create my own primers.
Yes.
I mean, that in itself like took weeks to do.
That was a pain.
Yeah,
Yeah.
so,
Yeah.
that's why I'm like, this is, I mean, I believe in this technology, if we get to work all the kinks out, so.
Yeah, we've been working on developing this for several years. We have just launched at WVC in February this year to offer it as a service. But yeah, the technology, all the background has been for years.
Very, very excited about data. It's just also it's the first time you're using NGS technique. That you first time get a full picture of what's going on in the sample.
Right.
Because previously you are just using culture, cultivate some organism that you can culture. It's the first time, and because we are the first time applying this technique here, and also because our technique was really well optimized, and for example, a lot of other sequencing platforms, they can only go to genus level resolution. Our technique can actually go to the species. This is critical. This is one of the obstacles that prevent us from entering the diagnosis field, like several years ago. After we tackle that, we think it's time. Again, it's the first time you look at those kind of data. And some of the data, like we basically look at skin and ear infection, and you will see we find a lot of organic pathogens that some of the pathogens that you mentioned earlier, those are anaerobes. And we talked to the professor from Lynn, right? So she said, I've never seen of that being reported. And we see it was highly enriched in infected ear samples. Those are anaerobes. And we found out those anaerobes are actually very important, very common as well, very common, very important. And they're also present in samples that have severe infection cases, like what I mentioned earlier was, those are probably indicated by a film. Because anaerobe, you see a sample that has a lot of anaerobes. That means it has some kind of protection. Otherwise, anaerobes, you exposed to oxygen, they cannot survive. When you have anaerobes, that means there's a biofilm or others protecting. That
Or the
actually
earwax.
indicates those are severe cases. Yeah. We are very excited about that, and we are thinking about public all the data.
This is so
Yeah.
Yeah, we're currently working on publications to have this in peer-reviewed journals as well. But
From a client perspective too, as you were saying, I was sending urine for culturing, and I still start them on antibiotics.
Absolutely.
And then
Right.
the others would be like, well, why didn't anything grow, but the antibiotics seem to work? And then they're kind of almost upset that they spent the money on the culture.
Don't you
when
hate
could have
that?
just given them the antibiotics.
And then you ask them how they're doing, well, it's doing great on the antibiotics, right? Yeah, it's frustrating.
We actually have a case of the urine sample. The urine sample is some of cases we found that actually mycoplasma was quite common there.
Yep.
And that's an infection, and that's a mycoplasma because it doesn't have cell wall.
Right.
Some of the common, like beta-lactamase, doesn't work for them, right? And that would be even a problem if you don't know what organism you're dealing with. That
And there's also been a few bladder, that'd be interesting pulling in a cap that I've seen that maybe you could tell me about. When you ultrasound it, it looks like there's a mass kind of bubbling around, and now that I'm thinking about it, maybe it's a really severe biofilm that's just
Yeah.
built up and it's just floating around in there.
Well, the
You
other
think
thing,
it's a mass, but it's not.
I'm thinking you ultrasound those, you get blood clots in there, the blood clots sitting there for a long time, and they hold the biofilm in there.
Uh-huh, yeah.
You know, or what about these animals, these cats and dogs that have bladder stones, and you take out, you take out the stone, and the bladder walls are this thick.
Mhm.
You know, then you go and you culture the bladder wall and you get no growth.
Right.
You know, and you know there's something going. There's more than just a stone going around there.
And where's your lab located?
In Irvine.
So it is in Irvine.
Yep. Yeah, we're just a few minutes from you on Alton.
I'll take some more kids then.
Absolutely.
Yeah, we've been, like our hospital, we've been collecting samples for like 3.5 years, I think. And I mean, for a while, it was about everything that walked in the door was sample. You know, whether it's pathogenic, and we separate out, this is the pathogenic sample, this is the normal sample. And we did it on so many different animals.
Including one bird was snuck into the mess.
Yeah, I do mixed animals, so I do on the bed at North Pike Fair also. So I do a lot of livestock, and so I sneak some of that stuff in too. You know.
And we come back, Dr. Mike, this is a weird dog. This doesn't look normal. We say, oh, okay, well,
Well, I mean, it's,
test passed.
I did that a lot just to challenge it, just to see what I would get.
Right.
Just to see it.
Yeah.
I'm a tester because I remember when I first bought my clinic, we were sending cultures to Antec, and we were getting a lot of no growth. And we had a specific driver who was putting all the cultures under ice, you know, and he was basically killing off all the pathogens that were in there.
Yeah.
And that goes along with this technology also, is, you know, you don't have to worry about transport, you don't have to worry about the way they're sampling. I mean, once it's in there, once you put it in the solution, it's frozen in time.
Right.
You know, you don't have to worry about human error from there to the lab. You know,
What that means too is frozen in time is from the time and site of collection. You're
Oh, yeah.
sampling the community dynamic.
Oh, yeah.
So it's pretty neat.
It's not gonna replicate it.
No.
Little test tube like
Yeah.
waiting to be tested.
or die. You know.
Price wise, what is it, money these days?
$80 per sample.
That's for one sample, but if you send in like an ear and a urine
That
and all
is
that,
per
it's 80 per site.
Yes, per set.
Not
Yeah,
per-
because they are
not per
separate
patient,
tubes.
but-
Yeah.
That's what I figured.
We do offer bulk discounts if it's a high number, or if you send multiple samples at once, so we can save on some of the transportation fees. That would help too.
And what's your turnaround time now?
2 to 5 business days from when we receive the sample. So, and then you get the report via email, so you get the results right away. It's a clickable link, and it's then a PDF. And like I mentioned, everything is clickable, so you can learn more about each organism that you find there.
And when you're doing it, are you still sending off the culture as well, just for a backup? You
No,
kind of relying on
No,
those
I
now?
just sent 2 samples in the other day, and both of either one of my cultures.
Yeah.
Only because now,
That's what I'd be thinking too. This
now
is
I'm,
superior.
now I'm trusting, you know, I've done enough cultures versus sequencing that I've seen that they're extremely compatible. What I'm seeing is they're exactly the same, except the sequencing is giving more information and more data,
Right.
you know, and finding things that the culture's not.
And you folks are only doing veterinary, you're not getting on the human side.
Only veterinary. Yeah.
You need capital investment. I think that's kind of some of the questions I have. I'll probably think of something, but. I think
Yeah,
I'll
absolutely. Give us a call or email. Either way, always happy to help.
You guys are always welcome to call me, usually always at the clinic, and I'm happy to talk about any of this stuff. You know, we also did, nobody mentioned this because we're still in the testing phase of it, but we also did we swapped colons on dogs, looking for normal flora. And I'm gonna tell you what, you should have seen the variety of that, especially with the diet. It was
Well,
extreme.
You know, our clinic uses IDEX and we do the fecal PCRs,
Right.
you know, and upper respiratory, and, you know, that only comes back with like 12.
Yes or no,
Yeah.
and it says yes or no,
Yeah.
you know.
Yeah.
I actually did that on a dog that was like a chronic kennel pup dog, I thought it was. And I swabbed that dog, but I also sent in a, respiratory PCR on it. And it came back, I mean, the whole PCR came back with negative, including mycoplasma. And when I sequenced it, it actually had quite a bit of mycoplasma in it. So now that can possibly be normal, but why did the PCR sequence come back negative?
Right.
And this one didn't. So I sampled it again with these guys, and it came back positive again.
So are all these primers proprietary for the company?
Currently, yeah, we are working on patenting, and a lot of research went into this, of course.
And then I looked at how because I use Antec, and I looked at how Antec was doing their PCR
Right.
versus how these guys are doing
Yeah,
it.
that's
It's like
wondering.
it's night and day difference. They're using technology from. I mean, it was the same technology I was using when I was an undergraduate.
Yeah,
You know, that's what they're using right now. And these
that's
guys,
what I'm wondering, if
Yeah.
they're just not amplifying because
right?
they have inferior primers.
I mean, it is a long workflow, everything from sample collection and the way you extract the DNA, and everything can have biases. And so we really optimize every single step of the workflow. Also the chimera removal, when you do the sequencing, the database is so important. The publicly available database is full of duplicates or badly sequenced samples, because anyone can upload anything. So we really went in and curated our own database, added our own organisms to it. It was quite a long effort.
There's a huge amount of state-of-the-art behind the scenes and
Yeah.
compared to culture.
Yeah.
And the beautiful thing is there are 3 manuscripts currently on new technologies and algorithms that were derived from just MiDOG. the MiDOG project. So dogs are leading the way
Yeah.
for novel discovery as well. So it's fine tuning the microbiome system.
Right.
Yeah, and we have all kinds of interesting samples. Like I mentioned, some dolphins. Most of the sequences are just unknown because no one ever looked at it before,
Right.
and it's just a whole new world. So
Here's an idea for you. You figure out what the healthy biome is and put it into a probiotic that they sell.
right, well, no
That's
single
actually
probiotic.
good.
I study
Huge investigation.
Yeah.
Shampoo or
I studied probiotics a long time during my PhD, with the effect that I'm not taking a single one of them.
oh really?
None of them are approved by FDA or EFSA,
Yeah.
so a healthy individual is no need.
What about transplanting the biome,
Fecal microbiome transplant, absolutely huge thing.
but also like for ears?
Ear microbiome transplant.
See, we've
Yeah.
done livestock, we do organ transplants, we've done that quite a bit,
Yeah,
you know, for a long time, right, and it works really well. But that's something I've thought about too, is the ear, and even the skin.
There's that product that I just heard about myself, Odic Armor. Have you heard about that?
Mhm.
The Odic Armor is something just now started being sold that you put in the ear, it's supposed to prevent infection for 3 months. And so I'm like, well, what are you doing there? Are you sealing up the skin so that nothing can grow? And how is that going to affect your testing too?
Right. Right.
Yeah, a treatment definite.
Yeah.
As long as the
It says it's
bacteria
not
respond to it, we will
steroidal
see an effect. Yeah.
and not microbial.
Mhm.
It doesn't say what it is.
Of course they do.
Maybe
Ah.
it does somewhere. I just kind of quickly glanced at it.
But yeah, but then it goes back to, I mean, you're talking about all that, all the probiotics and all things like that. You know, we see these dogs, they come from the groomers and, you know, they flush their ears every time and they wash out all that bacteria that's normal
Yeah.
flora. And these dogs are getting chronic ear infections, and it could be from that,
Yeah.
you know, and so the earwax has a purpose in that.
Mhm.
You know, he was talking about a lot of the anaerobic bacteria, and we
Right,
have a
right.
purpose. Now we're figuring that out. You know? And it's funny, we get a lot of criticism because they say, oh, it's contaminants, you have all these contaminants in there, you have all this other stuff in there. I don't necessarily think so. From all the data, all the cases I've seen, I don't think so. I think it's accurate. I think we're finding out new stuff, and I think we still have a lot of room to figure out what to do.
Right.
And once we do that, then we can figure out how to treat them.
And healthy dogs frequently have pathogens present, but very low amount,
Right,
because
right.
the rest of the microbiome is keeping them in check if you wash them away.
So maybe not a pathogen after all.
Opportunistic.
So a shampoo that has all sorts of bacteria in it.
Yeah.
Probiotic shampoo.
You know, you are taking on some big.
Yes.
Really, that's what some groups are looking at.
The other thing is we also recently found out, she also mentioned that in the presentation videos, culture tests, a lot of results from culture tests is only coming from 1 or 2 or maybe 3 single column. Using that to present the whole sample. Because culture, you just cannot allow to go deep. The way you guys know the culture is do a test is based on the single colony, and
Right.
all those cultures they use are expensive. They only allow based on how much they charge. They only test a few colonies, and using that to represent the whole thing. Rather than NGS was sequence and really deep into that, being able to tell you exactly different phenotype or genotype that's inside. Very detailed. Even if let's say there's a resistant microbe is present only in 2%, culture may not be able to pick it up, but NGS will definitely no problem to pick it up. And that's what we recently found out, and we're thinking about publishing data.
And so now, Carol, when your cultures stand here, because some dogs are so violently against it that you only get the external portion, are you really trying to effort to get as deep as you can then?
I am.
So would that even say?
Because it's
sedate
different,
them in order
because-
you can get
Right, because
a external is
right.
gonna be.
yeah,
I think,
well, it's
different.
it's different from the horizontal canal to the crown,
Yeah.
you know, because we've sequenced both the crown and then down in the horizontal canal, you know, the
On the
best
same dog.
on the same dog, and you'll see different floras in both of those on
It all
both.
makes sense. Yeah.
Right, yeah, because it's more of a
It's
closed.
vicious.
Right,
It's
exactly.
vicious all the way down.
So we've actually done that on some of the cases, sequence the ground and on the same dog. Once we've done that, we've gone down deep into the horizontal canal with the dog sedated, so we can get as clean a sample as we can. When I sedate the dog and do something, it's like, oh, here's an opportunity.
Okay.
I hate to say it, but we do it that way.
Yeah.
And I think that gives us the best data. But there is a difference between, next to the eardrum and outside by the crown. And I think, when you're sequencing, doing the area of infection or areas of lesions, I think is the best thing. But I know those dogs are difficult sometimes, but, we can only do the best we can.
Right. Yeah. And so if my techs are doing this and they accidentally, training on techs, if they get their own fingers on there, there's not going to be enough bacteria off of their fingers that
We might see some human organisms as well. So if it's a swab that looks like a Q-tip, anything you'll touch with it, we will pick it up.
I
But
need my sampler gloves.
yeah.
I've heard twofold. Number one, I say, I don't want you to get the infection. Number two is I don't want them to contaminate.
Right.
When we connect some healthy samples, we do see some of the ticks are. Because the ticks are, that on skin is very different from ticks on dogs.
Yeah.
We do see some ticks are has to be components.
Right.
Human skin and also our communication is probably something.
Yeah, regardless of what you're doing, sampling is so important and it's hard sometimes. So, and getting from the center of the lesion versus the outer part of the lesion, we've seen differences in that. Culturing, I think we get more no growth when we get the center of a necrotic lesion versus, the active area on the outside. I think that's kind of key. But it's not any different for the sequencing. So.
And our technique is very sensitive, so it doesn't care whether or not it's coming from a bird, from human, from dogs.
Right.
It just sequences all the microcells there.
Oh, I know how sensitive. I started doing experiments back in my day.
Yeah.
Contamination.
I think everybody does. Any other questions? You guys are real quiet over there.
Sounds very interesting.
Any questions? No?
If we do a culture and we have questions about results and we need to consult with somebody over the phone, for example.
You're welcome to call me.
Okay.
Yeah. If I'm not in the clinic, my staff can get a hold of me. Unfortunately. Or, you can call them and there's a couple
We
of
have a tech support on
Yeah, so you definitely have someone to talk to. So, and like I said, I've treated hundreds of dogs. So.
Okay, well, if you would like to take some sample kits home with you to try, please feel free to. We have them lying over there.
And then please fill out your evaluation forms. So those would be free to the left for us to submit, or you still charge us the $80?
We have 5 for free for the first time. Well, you can try again. So the first 5 are free, completely free, so you can see what the results are, how does it apply to your patients. You can even send it to culture and to us to see the difference. Give us a try, whatever you like. And then afterwards, it's $80 per sample. But
You just need to call, open an account.
it's very easy. You either go to our website, there's an order form, or you give us a call, or you give us an email and we send you the kit. The kit itself is always free, so you can stock up as many as you want, so you have them. And then you send us a sample, and at the end of the month, we count how many samples you send us, and then you get 1 bill for all of them, and you're charged on a net 30 basis. And 5 free.
You said something earlier about needing a larger volume of urine. How much
Right.
do you need?
Ideally, we would like to have at least 2 ml. I know it's not always possible, especially for cats, but so we have a urine kit specifically for urine. So the amount of buffer is very small, so the urine is not further diluted. So please use that if you can. And if you give us less than 1 ml, we will still try our best, but then it's like no guarantees, but at least 2 ml, ideally. The more, the better, of course. And so we have the swab kit that's really for any kind of sample, whether it's feces, skin, ear, anything, and the urine kit is only for urine. Okay.
Thanks.
Thank you.